seek into my favorite line of teaching. Viruses are small, except when they're not. Viruses are parasites. Viruses are simple, but complex. When I walk through my lab during the week cursing my fate at having to teach this course, and I yell at my students, they're really kind of scared of me this semester. They're just bags of nucleic acids. They're all the same. They're not really, right? We've seen some of the complexity that the flu paper with the host immune defense defeat mechanism. There are bags of nucleic acids, but as we've seen, they've evolved into some pretty intricate entities. By the way, we're talking about chapter 21, viral evolution of viruses. So, 5 years from now, let's say 10 years from now, when I come into your emergency room and you're on call, and I'm violently ill with some sort of infection, I want you to tell me as I go into it. They're small, they're parasites, they're simple but complex, and they evolve quickly. Andrew, I hope I don't roll into your emergency room.
Me too.
It's good with that. I hope my dog doesn't either. Not because of you. I realize that might sound bad, because a $1,000 vet bill, in my opinion, means a goodbye dog. A wife and kid disagree. We got a dog a year ago. He's an adopted. He's a pound puppy. Just so happy to be out of the pound. He defeated our backyard fence like 10 times in a row. Would run all over the neighborhood, terrorizing everybody. I live next to the Marietta Garden Center. He actually crashed a wedding there. We got a little call because he has a microchip. We went and picked him up, and like the ring bearers had him on a leash. So we have this picture of bride, groom, groomsman, dog. So we got a quote for an invisible fence. I'm like, that's insane. It was $1,000. I'm not buying that. 2 days later, he got hit by a car. He's fine now. $1,000 vet bill. God, the invisible fence cost me twice as much as it should. Anyway, so I'm way off on like dog and an emergency. I'll note nobody's asleep. That's a victory. So they evolve quickly. So this is the take-home point of the section on evolution. Why do they evolve quickly? I'm just going to steal my point. They're bad at replication. But that's good. From the perspective of an individual virion, it's not so much. They don't have perspectives. They're bags of nucleic acids. From the point of adaptation and continuation, the survival of the genes, which gets at basic evolutionary theory, that's a nice strategy, as we'll see. Why is it a nice strategy? Because those mistakes, the mutations, have a very low cost. What's the cost to you and me of a mutation in a gene? Well, it may well be death. And for an organism that has our reproductive and survival strategy, that's a downer. But if you're a virus and you're producing 10 to the 10 particles per day during an infection, an error rate of 1 in 1,000, as you'll see in a bit, that's not so bad. You can live with that. Occasionally, it's good that the virus gets a new quality. So those rare, advantageous mistakes aren't really rare. If it's a 1 in a million chance, I'm not going to put $10 on that. But you have 10 to the 12 events. 1 in a million happens a lot. 10 to the 6 times, 10 to the 5 times, something like that, a lot. But now you can go to sleep, that's all I'm gonna tell you today. So, where do we start? The whole universe was in a hot, dense state. And nearly 14 billion years ago, expansion started to wait, began to cool, the autotrophs began to cool, the developed tools, we built the wall. Math, science, history, examining the mystery, it all started with the Big Bang. Right, so we're going to go back to the beginning. That's the first time I've sang in class in about 5 years, and I think you see why I don't do it anymore. And actually, for reasons that escape me, I read the news stories. The Big Bang Theory in the last 2 weeks, not the TV show, the actual theory, got some serious experimental support that, according to news stories, is likely to win a Nobel Prize. But why that is, I don't know. So that's 14 billion years ago. And the Earth began to cool. 5. There we are, 5 billion years ago. 4 billion years ago, as we see from your textbook, you're starting to get cellular life forms. Bacteria, archaea, we used to call them archaea bacteria, but they're very different from bacteria. These single-celled, layman, single-celled prokaryotic life forms. But before that, in between Earth cooling and bacteria, we had to worry about, abiotic synthesis of organic molecules. And while my daughter was vomiting, I pulled this out, so it's not the word. So, theological implications aside, does anybody know, anybody who's not had biochem heard of the Miller-Urey experiment? No, good. Well, bad, I mean, that's horrible.
I've heard of it.
Good, good. 2, no, 3. No, you're just scratching your 4. So 1953. There's theories as to how life arose on Earth. Again, we're talking mechanics, not reasons why. There were inorganic molecules, so at least from volcanic activity, we get carbon dioxide, hydrogen sulfide, nitrogen. Sulfur dioxide, ammonia over the cheap seats.
Ammonia, nitrogen.
Yes. Yes, all right, stop stealing my thunder. What are we made out of, right? What are organic molecules?
Carbon.
CHOM. The worst acronym in biochem. CHOMPS. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. So the dirty little secret they don't tell you when you take Chem 3361, 3362 is that it's all irrelevant, except for these 6. So the sources of these were there, but as those of you may know, for example, nitrogen gas is inert. It's not mobile. You can't make stuff out of it. Unless you happen to be a bacteria that fixes nitrogen root nodules, but biological availability of N2 is 0. You have to have it in things like ammonia and then other sources of biologically available inorganic molecules. The atoms that are there, of course, protons, and it's one missing, nothing. So these things were present in the early atmosphere, or ocean, also in the atmosphere. Miller and Urey in 1953 set up something that, well, set this up. This was a prebiotic earth on the lab bench. They have water, heat source to simulate evaporation. The ammonia, methane, of course you have protons because you have water. Water vapor, of course being brought by the heating / evaporation that's not necessarily boiling, into a chamber where there's an electrical discharge between electrodes to simulate lightning, condensation, a little water jacket here, and then a pool. From this experiment, they generated, so this was ironclad evidence. So they got mostly glycine, but in that mixture they found 11 of the 20 naturally occurring amino acids. So no metabolism necessary, nothing building anything. This is spontaneous, well not spontaneous because you're providing energy, but this is chemistry that can happen in the absence of biomacromolecules. Interestingly, sugars were also present. So sugars are carbohydrates, so you really need carbon, hydrogen, and oxygen. What you may or may not know is that, so amino acids have an amine group. The amine group contains a nitrogen. The nitrogen has to be from a biologically available source, ammonia, but that ammonia comes into metabolic pathways now. The nitrogenous bases of nucleotides derive from amino acids. So it's a hop, not quite a hop, skip, and a jump. It's millions of years of evolution of the living force, but you can, in theory, get from amino acids to the nitrogenous bases along with sugars. And there's a pathway here to ribose, leading us to the idea that from the early atmosphere, certainly organic molecules could have arisen. What became biological molecules could also have arisen. So they did not get, by the way, nucleic acids from this. So there's a leap of faith here.
Did they get any lipids?
Lipids?
Yeah.
To my knowledge, no. My guess is absolutely not. But I'm going to go look that up. Thanks for asking. We get to something called the RNA world. Just me, or is this class not dragging on like most of them? It was the dog story, wasn't it? Yeah. I should give more tests. And this idea is that RNA would have been the first biomolecule to arise. For what reasons would you think? RNA, so let's talk about support for this theory. By the way, this was first proposed by Walter Gilbert in the 1980s. So RNA is capable of being catalytic, right? Ribosomes. So perhaps, what would that catalysis be? Its own replication. So this is my pre-central dogma over here. So we know RNA is capable of catalyzing reactions in a way that DNA is not. DNA is not a catalyst. Proteins can't code for their own replication. RNA is most likely suspect. I was supposed to have another order here. The Miller-Urey experiment shows we can get the building blocks from non-organic molecules. Anyone else? Yeah, so it's the information storage module. I'm kind of going that way. Protein, that's another way of saying protein doesn't really store the information necessary for its own replication. I'll agree with Brandon by thinking that we just said essentially the same thing in two different ways. Think about the central dogma today. Why would RNA have pre-existed DNA?
You needed to you needed to translate the proteins. You don't do that for DNA. You just do it for
That never happens. But I've already put this up here. Let's make it listen. Ribozymes as opposed to catalytic RNA, we have RNA protein hybrid enzymes. So where the RNA is playing a catalytic role. What am I talking about there? Sorry. The ribosome is the so-called smoking gun of the RNA world hypothesis. The idea being that RNA gains a function to encode protein. That protein does not encode the RNA, but along with the RNA forms the enzymes that more efficiently create this cycle. So RNA with the ability to encode protein is going to have an evolutionary advantage being represented in generations. Again, we're not talking about organisms here, we're talking about mere molecules. That an RNA protein complex is going to more efficiently encode RNA. The ribosome is the smoking gun. Of course, it's not encoding new RNA, but rather RNA and protein encoding RNA. The interesting fact of the ribosome is that there is not an amino acid within 18 angstroms of the P site. So what does that mean? That means the RNA in the ribosomes, a ribosome is a massive, multi-molecular RNA protein enzyme complex that does translation. The P site is where the peptide bond is formed between the two amino acids in the ribosome attached to the tRNAs. That's done entirely, effectively, by the RNA. So there's not any part of the protein that's directly responsible for that catalysis. Of course, the protein is responsible for the structure that allows for the efficiency there. But that RNA protein complex exists, and it's an RNA that is the effective catalyst, shows that we've got this factor arising, not with respect to replication of RNA, but polymerases also will have that sort of neat primase. Well, proteins are involved in replication of RNA and DNA, but we have this backing up long before we need the DNA. And of course, the DNA would arise from some stabilization of the information in the RNA. So the idea is we go from RNA to RNA protein to DNA coding the RNA coding for protein. So the central hypothesis comes out in that way. I'm missing one thing. Other metabolites, ATP, NADH, missing acetyl-CoA, are all derivatives of or nucleotides themselves. So that ATP that's our universal currency of energy in the cell, NADH, which is a coenzyme that allows for reductive biosynthesis. You'll see more of that when you think about biochemistry. And this is also involved essentially in oxidative or aerobic respiration. Those things that come later are built not from protein but from nucleic acids. So that's evidence for the nucleic acids predating proteins. What does this have to do with viruses? Well, viruses are like those organic molecules relative to the cell. So we think of viruses as, viruses are less than cells. They perform some of the functions of cells, but not all of them. So we have, with respect to viruses, a chicken and the egg question: which came first, the chicken or the egg? And I'm asking.
Well, parasites that kind of exist without cells to be frozen.
So which came first?
Cells.
The chicken. And it doesn't really work, right? Because the chicken lays in it. But in my example, the chicken, right? Yes. So Drew gets the point. He just didn't say chicken. They're parasites, so they can't possibly exist before the cell. That's not to say something viral-like did not. Our definition of a virus is that it's a parasite. You can't be a parasite unless there are hosts around. So clearly, cells predated viruses, so we can't have a virus until after the emergence of what we call living cells. Bacteriophages are almost surely much older than eukaryotic viruses. So, where did viruses come from? So we have candidates for viral origin. Hopefully I can tie it back into the RNA world here. One, messenger RNA. What is the purpose of a virus? To propagate itself. How does it do that? By encoding the information it needs to propagate itself. That means proteins. So even in prokaryotic cells, it doesn't show messenger RNA. We have messenger RNA here or there. That could be directly translated into protein. If you can find a way to propagate the mRNA, you then have stored the ability to encode the protein. So this would be plus strand viruses. As you can see from the figure there, the other two are transposons and plasmids. None of these encode enough information for replication of the cell, but they all encode some function. So you're taking for the example of mitochondria and chloroplasts, for an example, the generally accepted as fact now, mitochondria and chloroplasts used to be free-living, free-living prokaryotes. Just a quick review for those of you who haven't had cell biology in a while. Mitochondria is the respiratory center of the cell. This is where the electron transport chain happens, so all those products of glycolysis and the Krebs cycle are used to pump protons across the mitochondrial membrane to synthesize ATP. So mitochondria are the ATP generators of the cell. They don't do anything else except that they do. So mitochondria have their own genome, well, it's not really a genome, have their own DNA encoding their own genes. They look like bacteria. Chloroplasts are very, very similar, except that they're not. They're in plants. They do a lot of reductive biosynthesis. Photosynthesis looks a lot like the electron transport chain, except it's different. They all produce coenzymes and pass electrons down to generate fuel molecules. But as an example of functional symbiosis, mitochondria exist within the cell, and now part of our cells. They provide ATP, in return they get an environment in which they can continue. So we think of going the other direction. We went from prokaryote to organelle. The idea of viruses is they went from organelle, perhaps, virus. So you can imagine a lysosome, for example, losing function and not being a lysosome anymore, but rather being a ball of phospholipids. The example from your text, Mimi virus has a 1.2 megabase genome, has metabolic genes. Doesn't need them, it's a virus, doesn't do metabolism. But something that has broken off, lost function, but retains the ability to replicate when it infects. I'm sorry. I got something very wrong here. Example of loss of function symbiosis. So mitochondria can't live, are not free living anymore. They've lost a lot of their function. They retain function that benefits us and in turn similarly benefits them. It's like a parasite that's no longer kills its host. The difference between parasitism and symbiosis is a narrow one. So it gets right down to it, where are the viral origins? One, perhaps, molecular precursors. To go from RNA that encodes a protein that somehow gains a capsid, molecule up approach, which I've already erased, and two, cellular precursors. Perhaps some organelle or organism that has lost functions and therefore needs a host in order to replicate. Of course, there's one constant here. Oh, take home picture. Likely it's those, or of viruses that may have arisen from cell-free precursors, the single-stranded RNAs are the suspects. They're the most close to that RNA, world RNA, and of course, double-stranded DNA viruses are more likely to have arisen from cells or organelles, mitochondria that have lost function. That was tied to the RNA world. I was struggling to remember what it was. So the collection of all these ideas is that as your textbook says, we don't know where viruses came from. There are some ideas. One of the problems with viruses is they're not in the fossil record. You can't isolate dinoviruses. Viruses of dinosaurs, not some weird family of viruses. But there is the one constant worth mentioning, icosahedral symmetry. So regardless of single-stranded or double-stranded RNA or DNA, or double-stranded DNA or single-stranded DNA, every type of virus has members of the class that share icosahedral symmetry. Why is that?
You just have a repeating protein.
Why do they?
By low energy confirmation.
Resonance.
Resonance? Yes, that's exactly right, it's resonance. So it's pi orbital electrons. There's a reason why I've heard the idea. It's a solution to a problem, right? It's an effective, efficient solution with a couple of subunits. Some viruses have one type of protein that make up their capsid. You get a three-dimensional shape that's stable and encapsulates your genome. So it may just be convergent evolution that icosahedrons make sense for small infectious particles that don't have the necessaries to be alive. So why do viruses have icosahedral symmetry? I don't know. It works. So, the idea behind evolution, or evolutionary theory, is that genes perpetuate themselves. Those that do it better survive, increase their representation in future generations of population, and perhaps come to dominate. All of a sudden I feel like a coach during March Madness at halftime on ESPN. I want you to survive, increase, dominate. Well, my target was lost yesterday. I was absolutely certain the season was going to end based on a missed free throw, because that's kind of been.
I thought they were going to lose to Providence. That's
Well,
pretty sad.
Beating
Sorry.
Providence is not sad.
We weren't gonna do it right now. We'll talk later.
Viruses evolve quickly. Right, so one of the tenets of evolutionary theory is that random change happens over time that results in a change in the ability of an individual to survive. So it's selective pressure. Because viruses evolve quickly, because, as I said before, They're bad at replication. Particularly RNA. And retroviruses. There's RNA viruses, retroviruses, of course, have an RNA stake to them. RNA polymerases don't have proofreading, so this is another way of saying mutation rates are high. And then, two, there are selection pressures. For example, host immune response. I'm out of time. Remind me to pick up here Wednesday.